Today we have a lab demo for the Cryostat, and this is done by Catherine Lopez, a research assistant at Scripps Research. Could you describe the process as you go step by step?
Sure. So right now I have a brain on this mounting block, and I cemented it with a medium called OCT. It just keeps the brain in place. So I also have a very sharp blade right here that's going to help me with slicing the brain. And today we're slicing it for the PSTN, a brain region that our lab studies a lot. So we collect this section a lot. I'm just moving it closer to the blade so that I can begin slicing. And let's see.
So how thin are the slices that you take usually?
We do 35 micron, so it is paper, maybe thinner than paper, thin. Yeah.
And do you select, is there like a specific blade that you need to use in order to get the cuts that you want specifically?
So you need a specific steel Cryostat specific blade, and it's extremely sharp so that we're able to cut these tissue this thin. So right now you'll see there's a long trail of brain tissue. So I'm cutting through the olfactory bulb, and now I'm reaching the prefrontal cortex. So you'll see there's a whole train of just the prefrontal. None of this is of any interest for me to collect right now. So we're just slicing through until I reach the region that I'm interested in.
Okay. And this all ends up on a collection plate where you can just collect it and use it later on?
Yes, so they will be collected into little vials that look like this. It has a cryoprotectant inside of the vial, so we can store the tissue at a -20 temperature for long term. And depending on what we want to do with it, we may stain it, we may just mount it on slides and look at it, or keep it for future experiments. We have many old brains from like 10 years ago that are still in those freezers, waiting to be touched.
What do you do with parts that you do not use?
So with the parts that I don't use, like such as these, which you can see we're cutting through a lot of the brain, they just end up getting trashed, truthfully, because it's not the region that we're interested in looking at. So if it were the whole brain that I were collecting, then all of that I need to keep.
Okay.
Yeah.
So what do you do with the samples once you collect them?
So once we collect them, we'll put them in a storage vial, and we will, sometimes we'll stain them, sometimes we'll mount them on slides and take a look at the microscope with them, and see if there's any, native signal coming out from the PSTN region. It all really depends on the questions we're asking with the PSTN. So some of these brains, they were, before being collected, they were manipulated through surgery, injected with viral vectors that target cell-specific neurons that, while the mice are alive, we go ahead and do a lot of behavior tests. We do, for our lab, since we study alcohol, we put them through rigorous weeks of drinking alcohol, being exposed to alcohol through vapor. And then once all of that is said and done, we collect their brains, and then we look at that brain region to see like how was it affected after being through all of these trials of alcohol.
Mm-hmm.
So the withdrawal effect is what we're interested in. Yeah. Okay. Okay. So now we can go ahead and start doing the collection. Oops. That happens. Oops, sorry.
Oh, what happened?
So I collect 5 sections at a time, and 3 of them are on the blade, but 2 of them fell down.
Okay.
It happens. That's okay. As long as I know which one is which, then we're good.
And this is which part of the brain?
The PSTN.
Okay.
So that's called the parasubthalamic nucleus. There's a lot of research out there that this region may be an influencer on drinking behavior, drinking more, drinking less. What our lab is trying to do is use mice as a model to investigate this area and figure out what is it doing, which type of astrocytic cells are influencing this behavior, since there's a large population of CRH Cree and TAC1 Cree subpopulation cells that live in the PSTN, and they seem to have some kind of influence on how the mice drink. So that's what we're exploring.
And you use these mice as animal models to essentially think about human behavior as well.
Yeah, we always use animals as a model first, model organism, to, take a look at these questions and understand in a more microscopic way what is going on here. The cool thing is that these brain regions also exist in humans as well. So there is a little bit of maybe like you could hypothesize a little bit that if it's happening in the mouse brain, maybe it's also happening in the human brain. We won't know for sure, but you can kind of make that assumption after working in many mice and doing that over and over again. So I'm just going to label the slide with the ID, the brain region I'm looking at, and if it were stained, then I would put down what it was stained with antibody-wise. In this case it wasn't, so I'll just put my initials and then today's date. Tissue mounting. So the tissue is sitting in 1x PBS right now. It was washed off after sitting in the cryoprotectant from that vial.
Could you just briefly explain what tissue mounting is?
Yeah, so tissue mounting is when you take the tissue that was just sliced at the Cryostat and pick it up and put it on the side. So that's the mounting part. So in this case, what I'm doing is just taking a look at all of these sections. They kind of look the same at a glance, but when you look much closer, you'll see that the structure of the brain changes. Because as you saw at the Cryostat, we were slicing from the front to the back. And so in here, the sections will be the same, where some of them will be more anterior and others will be more posterior.
So you'll be looking for a tissue that has most of the parts that you want?
Most of the parts that appear in the beginning. So right now, this one looks like the first one.
So how exactly do you identify the tissue that you need?
So there's different structures on the sections that help clue me in to where I'm at. In this case, it would be this part right here is a dark kind of butterfly wing structure at the top half. That would be the hippocampus. It starts off small and then gradually it starts to wrap around and get bigger. So let me just find the next one. I think it would be this one. So it's really easy to tear the tissue because it's so thin. So you have to have a delicate hand when holding the brushes so that you don't rip the tissue.
So what happens if the tissue does tear?
Then I do my best to put it back in place when I know which section it was originally with.
So is there usually a maximum of 3 tissue samples per slide?
What was that?
Is it like a maximum of 3 tissue samples per slide, or could you have more?
Oh, you can have more. Everything inside of this petri dish, it can fit on the slide.
Okay.
Yeah.
So this is where we're adding a mounting medium to the samples, and then we'll be adding a cover slip in later.
So I'm taking about 50 microliters and I put it on the slide. And I'm going to go ahead and take my cover slip.
So just to clarify, the mounting medium is not a dye, or?
This mounting medium is an adhesive for the glass cover slip. It will protect the tissue and preserve it for later on when we store it in slide boxes. It also has DAPI in it so that when we take it to the microscope and shine a light on it to look at its fluorescence, the DAPI will help us navigate what we're looking at, since it will tag all the cells. And so this is what it looks like after putting a cover slip on.